This tutorial will focus on monohybrid crosses and how to do them, and we'll do this just by working through an example. So monohybrid crosses, as the name suggests, focus on just one particular gene. So the one gene that will be crossed between two individuals. And then it looks at the probability of all the possible outcomes from fertilisation. So let's take the example of just a common example of mice. And in mice we'll look at two phenotypes. We'll look at black mice and white mice. So the appearance of the mice are the phenotypes, and the gene that controls these phenotypes, of course, is the genotype. So let's say in this example that the colour of the coat of the mouse is under the control of a single gene. So a single gene, and it's a dominant recessive pattern of inheritance. That is, that one allele is dominant over another allele, the recessive allele. The first thing you need to do is to denote letters for the alleles of this gene. So when it says denote a letter, you can choose any letter that you like. Generally a letter is chosen to represent the first letter of the phenotype for the dominant allele, but it doesn't have to be this way. It could actually be any letter that you like. But in this case, let's choose capital B to represent the gene. Actually, we'll just change that to represent the allele for black coat colour. And you'll notice that dominant alleles are always uppercase letters. So it's a capital B always for the dominant allele. And let's denote little b as the allele for white coat colour. So it's important here to recognise that the recessive allele is always lowercase. So let's just try and imagine what we're looking at here. So when we talk about the phenotype of coat colour being under the control of a single gene, what we're talking about is in the cells of the mouse there will be a homologous pair of chromosomes. There'll be several pairs of homologous pairs of chromosomes, but on one particular pair will be found the gene that controls coat colour. And in this particular case, the gene for coat colour has two alleles, big B and little b. So a mouse could have the big B allele on both of the chromosomes in the pair, or it could have big B and a little b, so the heterozygous combination, or it could have little b, the homozygous recessive combination. They are the three possible combinations for the alleles in this mouse. Alright, so what we're going to do is we're going to cross, that is breed, two mice. One parent is going to have big B on its chromosomes, and the other mouse is going to have little b. So let's again just picture what's going on here. So this mouse is going to have big B alleles on its genes, and this mouse is going to have little b at the same locus on its pair of chromosomes. The first question we have to ask ourselves, actually before we do that, we'll just want to write in here that this is the parent generation. So these are the parents, often just denoted by capital P. And these are the genotypes of the parents here. So the first thing we have to work out is what the possible genotype makeup of the gametes or sex cells of each of these mouse cells is going to be. So we have to remember here what happens in meiosis, and that in meiosis each member of a homologous pair sorts independently into separate gametes. So half of the sex cells in this mouse will contain the chromosome with big B, and the other half of the sex cells, of course, will contain the other chromosome. In this case, each half of the sex cells, of course, will be identical because it's a big B, big B. So all of the gametes from this mouse will contain a big B. Exactly the same process over here. So each member of the homologous pair will separate during meiosis into separate gametes, and each half of the gametes will contain one of the chromosomes, each one carrying a little b. So in effect here, all of the gametes that are produced in this mouse will have the genotype little b. So these are the gametes that will be produced for each of these mice. Then the final question that we ask ourselves is, how many different possible ways could these two kinds of sex cells meet each other in fertilization to produce offspring? In this case, all of the gametes in this mouse have a big B, and all of the gametes in this mouse carry a little b, so there is only one possible combination for these two. So every mouse produced from this cross must contain a big B and a little b. So the genotype is big B, little b. 100% of the offspring will be big B, little b. This is the genotype. And the phenotype, what will it look like? Of course, will be 100% black coat colour. Okay, so that works through the cross in a bit of detail. This cross could be just as quickly summarised in using a Punnett square, so let's have a quick look at how we do that quickly. So our parents are big B, crossed with little b. These are the parents. The gametes, all of the gametes in these mice will be big B, all the gametes in this mouse will be little b. If we were to do this as a Punnett square, it's very straightforward. Whoops, got that wrong there. So this is a very simple cross. All of the offspring are big B, little b. So let's have a look if we were to do a different kind of cross here. Let's say our parents are big B, little b, crossed with big B, little b. So they're both heterozygous parents. So what's going to happen this time? So in this case, remember these are on separate chromosomes in a pair. So half the gametes will contain a big B, and half the gametes will contain a little b. In this mouse, half will contain big B, and half will contain little b. So here we can set up a 4x4 Punnett square, and we put the gametes, possible gametes, along the top for one mouse and down the side for the other mouse. And then we would say, well, if this gamete here met this gamete here in fertilization, then the offspring would be big B, big B. If this gamete here met this gamete here, then we'd have big B, little b. If this gamete here met this gamete here, you'd have big B, little b. And if this gamete here met this gamete here, you'd have little b. These are the only possible combinations of the gametes in this mouse. So here you can see that 1/4 of the genotypes are big B, 1/2 of the genotypes are big B, and 1/4 of the genotypes are little b. Or you could write this as a 1:1 ratio for the genotypes. And you can see that 3 out of the 4 mice, so 3/4 of the mice, or 75%, will have a black coat. And you can see that 1 out of 4, or 1/4 of the mice, 25%, will have a white coat.